Hello everyone, I'm called Mariam and I'll be voicing today's presentation titled Steps Taken to Develop an Optimized Texture Analysis SOP for Boiled Sweet Potato to Predict Sensory Texture. I am presenting on behalf of a team of very elegant scientists who contributed to this work at different points in time and also brought different expertise. This is an outline of the day's presentation. To begin, I take you back to 2019 when we were able to still meet in person, and Layal presented these to show the steps you need to follow when developing an optimized method for instrumented texture. We have made a few modifications because we believe that statistical analysis is a relevant component, is a guiding factor when making the choice at every stage. But we've also removed the evaluation of optimal cooking properties because our intention is to develop a method that correlates with sensory texture and not necessarily cooking properties. Also, you will see that based on this diagram, the procedure is presented as a stepwise cyclic process. However, in actual life, in practice, we found that there were times when we had to do two things at the same time. So the presentation here is merely for simplicity, and you may find that your approach is varies a little bit in actual practice. Again, in 2019, we had already recognized the challenges when working with sweet potato texture. First, the roots come in varied shapes and sizes. There's also variation in texture within a single root, but also between different roots of the same variety. So this means that we have a lot of heterogeneity to deal with. We had at that time already established a first draft for our instrumented texture SOP, where we made cubes of 2.5 centimeter size and immersed them in water at 85 degrees for 15 minutes before analyzing texture using a knife blade. There were things that were fine with this method, but there were things that were not so fine with this method. And those included that the sample preparation we were following for the instrumented texture was really different from the sample preparation method we were doing for sensory. Particularly, we were immersing the samples in water for the instrumental texture, whereas for sensory, we were steaming. We also developed this method by correlating with optimal cooking time, so it was optimized using the cooking properties. However, we needed a method that can correlate with sensory texture. So, reflecting on upon reflecting on our method, we recognize that these points, preparation procedure of the product, and correlation between sensory texture and instrumental texture were the most important that we had to focus on moving forward in our modification of the existing method at that time. So then we started off with our first set of experiments, and our first set of experiments was aimed at identifying a preparation method where sweet potatoes were steamed that gave robust and discriminative results. We used NASPOT 8 and NASPOT 11, which are contrasting genotypes in regards to texture, and prepared them in following three different ways, which I'm going to give details on shortly. For texture analysis, in all cases, we did TPA, double compression, 40% compression on pieces of this dimension. And for the texture output, we focused on parameters of peak positive force and area under the curve in both the first and second cycles. However, in the data presented here, and also in other experiments, I will be focusing on the results from the peak positive force, even though the trends were really similar among all the parameters. Just for simplicity, and to keep it short. And here are the methods we were using. Like I said, there are three. The first one, method A, which we now commonly refer to as the kitchen method, follows the same exact procedure we use for sensory analysis, to prepare samples for sensory analysis. And there we prefer portions of a particular size. So we have some level of control on the weight of the samples we use. With method B, which we now commonly refer to as the strict method, is where we exercise maximum control over the shape and size of the samples. We make cuboid portions of this dimension, and you will notice that this dimension is quite bigger than the dimensions of the 2.5 cubes that we used to make in the first draft of the instrumental texture SOP. When it came to method C, our thinking was, you know what, in real life we are bound to get some sweet potatoes that are quite big, that are outside the range that we prescribe or recommend for method A. Is it possible for us to somehow modify the size by cutting off some bits of the edges and still be able to get robust results? So that's the idea of method C, and we really, in this method, had minimal or the least control over the shape and size of the samples. On this slide, we try to give more details in regards to the texture analysis and data analysis. I would like to bring your attention to how we went about the data analysis. We looked out for two things, the first being reproducibility. And here we were wondering, are we able to group roots of the same variety together? So in this case, we conducted ANOVA to compare the means of the different roots of the same variety. And here we're looking for no significant difference between the means of roots of the same variety, and that would be taken as a reproducible method. The second thing we're looking at is discriminativeness. So is this method able to help us tell the difference between two genotypes that are different? And in this case, we were doing ANOVA on the means of the different varieties. And here we're looking for a significant difference between those means. This is a table summarizing the results. On this line, it's about reproducibility of NASPOT 11. And then on this line, it's about reproducibility for NASPOT 8. And on this line, it's about discriminativeness between NASPOT 8 and NASPOT 11. And you will notice that method A and method B were both reproducible and discriminative. Method C was not reproducible and not discriminative. So it means that method A and method B are the more preferable methods to take along with us. Another thing that you may not be able to appreciate from this table here is that we noticed that with method B we had many aborted readings, and we knew that even though it was giving us good results, that problem of aborted readings is something that we had to solve before moving along with concretizing it as a method. So those results informed our second set of experiments. And in our second set of experiments, we wanted to know, yes, we have been using TPA and the Plaid probe, and they've been working fantastic for us. Is it possible that there is a probe that, or another texture analysis method that would give even better results? And then the second question was, how do we modify method B such that we reduce that problem of aborted readings? We purported that the reason for these aborted readings was because the samples were a little bit hard because they had been perhaps undercooked. And how we went about modifying method B was, we first of all reduced the portion size slightly, and we also increased the cooking time. And then we also, when using the TPA method with the Plaid probe, we reduced the level of compression as well. Besides the modified method B, we also prepared samples using the kitchen method. Besides the TPA double compression, like I mentioned, we reduced the level of compression from 40% to 25% to deal with the issues of aborted readings we had noticed in the first set of experiments. We also used a cylindrical probe and lead penetration test on the samples. We did the same data analysis like I presented in the first set of experiments. On this slide, I just wanted to give more information in regards to the number of roots, number of readings, sample size, probes, program settings, and parameters that we used in all the different cases so that you'd have a better understanding of the different methods. This is a pictorial illustration. I mentioned that for the cylindrical probe, when it comes to the kitchen method, we conducted the test on vertically cut halves of samples for the kitchen method. For the cubes, well, we just used the cubes. For the TPA, for the kitchen method, we still had to excise, we still had to cut out portions of a similar size as the portions that we get from the strict method. And here is the table summarizing the results from that set of experiments. Again, this line is about reproducibility for NASPOT 11. This line is about reproducibility for roots of a jumula. And then this line is about discriminativeness for the between the two varieties. And as you can see, the strict method was always reproducible and always discriminative. Yes, the problem with the aborted readings was greatly resolved with the modifications we did to the method. However, with the kitchen method, you will notice that it was always discriminative, but not always reproducible. So it was half and half. And then one thing that you may not be able to appreciate on this slide, but from this table, but something that we noted was that with the cylindrical probe, the raw data was such a mess, and we had to do a lot of cleaning in order to get the data that the information I present here. Actually, one of our researchers, Christian, completely refused to accept the cylindrical method gives reasonable or useful or meaningful results because of that. And I think it's based on your discretion as a researcher. I mean, it takes a lot of effort and a lot of time to get there, but ultimately we would be getting there with the cylindrical probe. However, why suffer with all that strife when there is a Plaid probe method, which you may have to clean the data, but you don't have to do too much. There's not too much effort you need to do. So based on the results here, it was clear for us that it was best to use the strict method for sample preparation and the Plaid probe TPA double compression for texture analysis. Another thing we did was that we tried to identify the source of variation, compare the variation between the roots based on variety, probe, and preparation method. And here we saw that the preparation method was not really a significant source of variation for the texture differences between the samples. And for this reason, we can see that we concluded that the readings from or the results from both the kitchen method and the strict method of sample preparation were really consistent between one another. And so even though the kitchen method does not give the best results in terms of reproducibility, we identified it as a convenient alternative for the strict method. And this is really important because, like I noted before, the kitchen method is the same exact process we use when preparing samples for sensory analysis. So in cases where we are conducting both sensory analysis and texture analysis, it would be more convenient if we had the same preparation method. So, with following the second set of experiments, the next step was to optimize the method, the strict method, TPA, double compression, and see if it was really correlating with sensory, because that's the ultimate goal. And here we used 12 genotypes, and like I mentioned, we had chosen our method of texture analysis and our method of sample preparation. When it came to the results, the ANOVA results, you notice that with our 12 genotypes, we're able to get discrete groupings for the texture of the different varieties. And the data presented here is really important because here we used raw data. So before any form of cleaning, you can still already be able to tell, to distinguish, to identify discrete groupings of the different varieties, which demonstrates just how discriminative this method is. When we did the correlations between sensory texture and instrumental texture, we realized that there was indeed a good correlation between sensory hardness and sensory hardness, both by hand and in mouth, and parameters of instrumental texture, particularly positive force and positive area of the first calf. So which means that we had been able to optimize our method with sensory. Moving forward, we would like to still see if the kitchen method could still give us results that correlate well with sensory texture. Like I mentioned, it would be a convenient method, a convenient alternative to the strict method of sample preparation. The take-home message from here is one, that the development of an instrumental texture analysis is a process and it takes time. As you have seen, we have gone through several sets of experiments, and guess what? We still have to do some more. So sometimes you have to take 2 steps, you take 2 steps to the front, sometimes you take 2 steps back, sometimes you take many steps at a time, sometimes you have to take, small steps. And then the other thing is that the process requires a multidisciplinary team, because from the breeders we needed 2 varieties of contrasting texture, but also we needed very many roots from each variety, and they had to be able to supply those and advise on which varieties would be best to use. And then we also required different expertise within sensory science, within food science. So sensory scientists were important. Scientists who know a lot about texture were important, and then data analysts were also important. And it could be the team you require could vary in a different case. So that's all I had for the presentation. I'd like to extend my sincere gratitude to Christian, Christophe, and Layal. Christian advocated for this presentation to be made. Christophe and Layal literally coached me for today's presentation, and my supervisors, Mukani and Dr. Sally. I'd also like to acknowledge the contribution of NARO through providing their facilities. Thank you.
Thank you very much, Mariam, and thanks colleagues from CIP, CIRAD, GHI, and North Carolina. This is indeed a multidisciplinary team. Thanks for this very insightful presentation, which actually shows the whole process most work package 2 teams will probably have to go through at one time or another to develop a robust, reproducible, and discriminant protocol for instrumental texture measurement. So protocol which is correlated, which helps to correlate with sensory descriptors, because as Mariam mentioned in the presentation, this is the ultimate goal in our research. So feel free now to raise your hand to ask questions directly to Mariam, or to post your questions or comments in the chat box for Mariam and CIP colleagues from Kenya and Uganda. I see there are already a few questions from Thierry. Thierry is asking, what is the difference between Plaid probe and cylindrical probe? Does the Plaid one cover the whole sample and the cylinder covers only part of the sample? Mariam, how do you want to answer Thierry's questions?
Thanks, Thierry, for your question. So the Plaid probe is more like a larger surface area, and yes, it covers the whole sample. And the cylindrical probe is like the needle. It's like a needle, so it just penetrates the sample.
Okay, thanks, Mariam. What is the diameter of the cylinder probe?
It was 3 millimeters.
3 millimeters.
3 millimeters,
okay.
Yeah, for the cylindrical probe, yes.
Okay, thanks. Okay, maybe I had the next question is in the chat is for me as well. So I wanted to know how many repetitions you do per genotype, like how many pieces you have to do for one texture measurement.
So it depends on the preparation method. So for the kitchen method, we use about between 4 to 5 roots, and from each root we get about 2 to 3 measurements, but mostly we get 3. And then for the method where we make the cubes already, we only need about 3 roots, and from each root we get about 3 to 4 pieces. So 3 to 4 measurements. That is for the TPA.
Yeah, got it. So in total, it's about 10 to 12 individual texture measurements.
Yes, for the kitchen method, we get at least 12, and also for the same for the strict method.
Yeah, and that's good. Thank you.
Thank you, Thierry, for these questions. There's a question from Dominique. For population screening, how many texture measurements per genotype can you perform per day on cooked products?
Thank you, Dominique. We are yet to find out. We are yet to find out, but we hope that we can, because of COVID and we have to leave early, I hope we can do a minimum of 8 genotypes. And right now the only method we have optimized is the strict method. So we will most likely be using that. And that means that we will be using 3 roots per genotype. So we'll be having about 9 measurements minimum per genotype. I hope that answers your question. And we do all the analysis on cooked products for texture.
I think that answers the question. Thank you. Lots of questions in the chat box. One from Akim. What was the temperature during the analysis, and was it controlled throughout the analysis?
Thank you so much for this question, Hakim. Yes, we did control temperature. It was difficult to come to a temperature to use because very often we realized that with the sensory analysis in the beginning, the samples were being evaluated warm, and we could not maintain the temperature when we're doing the texture analysis. So what we had to do in the end was we resorted to doing the texture analysis on samples at room temperature. It was much easier for us to control that way. And what we did is when we are doing sensory analysis, we use CompuSense. So we put a time lag for the panelists before they start doing texture so that it allows the sample to cool down. So you find that the sensory analysis is conducted at more or less the same temperature as the texture analysis, so in that way.
There are 2 questions from Amos. I'm not sure I get the first one. How was the sensory evaluation done? How many sensory panelists did you use? So maybe answer the second one. The second one is more straightforward. How many sensory panelists?
So we have a sensory panel. Our sensory panel, well, the composition may change, but we have they usually come up between these about 13 people trained, and they come between 8 to 12 of them show up on a given day. And according about how the sensory evaluation is done is we already established a method, an SOP for sensory evaluation. So we follow that method. It's using a panel, a trained descriptive sensory panel. And those are the number of panelists we expect for each evaluation.
I hope it answers your questions, Amos. I'm jumping directly to Jim's question because it is about sensory. So were there other sensory attributes to which you can try to explore correlations, for example, mealiness, try mouthfeel, smoothness, etc.?
Yes, there were others. And one of the ones that we are following up on, we will follow up on, I think, is particularly meliness, because it's one of the more important sensory attributes coming from work package one. So our breeder, Dr. Sally, came up with some models, some tentative models, and they showed that it was actually possible to make some predictions on meliness. So right now we're mostly focused on hardness, but I see us in future trying to establish maybe perhaps models to predict other sensory attributes.
I hope it answers your question, Jim. Another question from Michael. Michael is asking, did you check different strains before using 25% as the most appropriate strain for the TPA tests?
Yes, in the first case, we used 40%. And then that's when, after we got so many aborted readings, that's when we decided to do 25%. And we got the problem of aborted reading sorted. So yes, we did try some others before settling on 25%. Thank you, Michael.
Another question from Thierry. What information can be obtained from the second compression? Did some of the samples break during the first compression? So 2 questions actually.
Wow, thank you very much, Thierry. Did some samples break? Like break apart? Yes, I did notice some samples break apart on the first compression, during the first compression, and yeah, or crack a little bit. Some others just got smudged. I'm not sure. I think it's something we could throw to Lyal, what information can be obtained from the second compression, because so far we were still able to get a little bit of information on hardness, but the information was not as good as from the first cycle. Yeah, but I think that's a good question, and it's something that maybe we need to think about.
Hey, yes, please.
Okay, thanks, Mariam. I was asking those questions. Actually, the second question, like whether samples break during the first compression, is it the same as aborted measurements? Like when you said the measurement is aborted, does it mean that the sample broke and then it was not possible to complete the measurement?
No, when I say aborted readings, I mean that because our machine has very few load cells, so what happens is when the sample is too hard, when it exceeds, when the force required exceeds what the instrument can manage, it aborts the reading.
Okay.
It stops, it does not complete the measurement.
Yeah, I see the difficulty. Yeah, so I still have that question on the information that can be obtained from the second compression, because, okay, deeply I have doubts about the TPA. For example, if the sample breaks during the first compression, then we don't have a good sample for the second compression, and it's not clear what the texture analyzer is measuring. So it's not clear what information we get from the second compression. And I've been looking in the literature for several years, and I haven't found a case study showing what exactly the second compression gives as information. So indeed, it's something to study further. Thanks.
Thank you, Thierry. A question from Toyin. What would you advise texture measurements on different parts of the roots? So anterior, median, posterior, let's say proximal, central, or distal part, or just the median or the central part? Because consumers, he mentions, consumers eat all parts.
Yes, I'm sorry, I cannot pronounce your name, but yeah, you're right. The consumers eat all parts. And in our case, besides the tips, we only remove the tips because usually in sweet potatoes, those in certain sweet potatoes just tend to be just a little bit fibrous. But we just remove those tips and we just remove just a bit of the tips. And then whatever is left in the middle, we cut out the portions from there. So I think we do not have a similar approach to the cassava team in that way, because they try to measure texture and compare the different parts, and we don't do that. We just remove the extreme bits, and then the rest of it we perform texture on all of it.
I think it also partly answers Wasiu's question. And Wasiu is asking, maybe as a compliment, don't you think the different part of the tuber may have different texture attributes?
We do know that, yes, that like I presented, maybe on the first few slides, that there is variation within a sweet potato root. However, unlike I think many of the other tubers, it's very hard. First of all, a sweet potato root is perhaps much smaller than, say, yam or cassava. So it's very difficult to tell where the posterior is, the anterior is, and where the middle is. But also the other thing is that the distribution of texture really varies from root to root. So you cannot say, I don't know if that answers the question. So it's just much safer for us to just remove the tips and conduct texture on the whole root. And I think that's what one of the objectives of setting up our method was we knew that we had a lot of heterogeneity to deal with, and we're looking for a texture method that could help us deal with that heterogeneity, that could help us still be able to group roots together of the same variety, even though that we know that there is quite a lot of heterogeneity within a sweet potato root and between roots of the same variety. So I think, yes, what you're saying is true, but we knew that going in, and we set an objective directly addressed to that. And right now we have a method where that is not so much of a problem.
Thank you. Dominique is asking, are you planning participatory trials? So let's say within work package 5, for which we would have consumer tasting, sensory panel, and instrumental texture measurement data sets. So possibly to establish limits of acceptability thresholds, consumer acceptability thresholds.
Thank you very much. I'd like to pass this question on to either Mukani or Dr. Sunny.
Okay, thanks, Mariam, for the presentation. I'll take that on. We have just established the on-farm trials with NACRI, and with that we shall have sites where we can conduct at least the consumer testing, and then back that up with data that we shall continue to evaluate those materials also in the lab for the sensory and texture analysis.
I'd like to ask maybe Layal, who has been involved in the preparation of this presentation and who has been supporting the CIP team in the optimization of this protocol. If she has a word to say, Layal.
Yes, Eglantine, thank you. Good morning, everyone. Mariam did a good job, so I don't have anything to add except saying bravo. And thank you a lot for the collaboration that we had together with the sensory team, the CIP team, and with the CIRAT team. It was a great experience, and I hope that the other partners will do the same, and I think we can make a very good job together. Thank you, Mariam, again, and thank you for the CIP team.
Thanks, Layal. Last question from Christian in the chat box. Do you plan biophysical analysis in parallel to texture measurements?
Yes, Christian, thank you. So yes, and we will be, once we get the samples ready, we will be sending them to your lab, I believe, to do some analysis on them as well. But we will be also doing some work with RVA to do some starch characterization as well.
Dominique made a comment regarding your textual meter in the chat box, but it's not a real question.
But he's right, his comment is right. So because we realized that it was not strong enough, we made those adjustments so that we could still work with our texture analyzer.